All right. Okay. Thanks for the message, Dr. Wood. Okay. Hello and welcome all to my PhD defense seminar. I'm so excited and thankful that people around the country today are willing to spend an hour to hear about the exciting science that I've been doing while a graduate student here at Duke. The past few months have been hard for so many people because of COVID-19, but I've been so inspired and encouraged by the way that people around the world have responded to this crisis, especially essential workers, parents, and students who have adapted to learning in a virtual environment such as this. For many of you, this is the first PhD defense you've attended. Typically, these defenses occur in person in an auditorium somewhere on campus and consist of two parts. The first is a public seminar, which is what you're tuning into now, and the second is a private defense where members of my committee will examine me before deciding whether I've earned my doctorate. Due to COVID-19, PhD defenses have had to become virtual, which creates an additional set of challenges. As many of you know, internet hackers have been wreaking havoc on Zoom meetings across the country. Therefore, we've taken some extra precautions to ensure that everyone has a pleasant and informative time today. So welcome to YouTube live stream. I'm currently streaming live from my home and will be giving the talk in this format. The only difference for you is that if you'd like to ask a question or leave a message, you'll need to do so using the chat box, and some people have already started doing that. A couple of things about the chat. First, we're using slow mode, which allows you to only type one message every 30 seconds. Because of this, please try and ask your question in one block of text. Second, comments containing inappropriate messages will be automatically filtered, and you may be silenced for the remainder of the talk. That's mostly for any trolls that might be in the audience. Anyway, with that out of the way, I'm going to turn it over to Scott for a brief introduction. Sorry.
Hello and welcome. Thank you all for coming to this PhD dissertation defense from Drake Edwards. I really wish that we could all be here together face to face to appreciate and celebrate the hard work that Drake has done putting this dissertation together. But I think in usual fashion, Drake has mastered another feat, and that is this technical virtual world that we're now living in. And I think the two stories that he will tell you today will really shine through in the presentation that he's put together in this virtual world. It's my pleasure to introduce Drake Edwards. As many of you know, Drake grew up in Iowa and attended the University of Iowa, where he was a physics major. And among his many impressive accomplishments as an undergraduate student, he had an invention. He was part of an invention team that created a radiation therapy device for brachytherapy that is now patented. And so that's impressive. And I think that it's his interest in radiation that initially led him to my lab, where there was a bit of a bait and switch because we don't do a whole lot of physics in my lab. Instead, it's really a biology lab. And Drake dove right into a rotation project. The end result will be, of which you will see as the first story that he will tell you today. And this was a pretty mechanistic biology project that Drake really tackled. And I could see him sometimes wrestling with biology in general and the fuzziness of some of the results. And it really kind of went against his quantitative, careful manner. But I think that's a really good quality that you'll see in Drake's work, that he demands quantitative and scientific rigor from projects. And it was this quality, along with his determination, that really led him through a lot of obstacles to bring this first story to near publication. We have really phenomenal reviews that we will very soon see this in print, I think. Of course, Drake has many qualities. One of them also is his skill with a scalpel, as I think you can all see displayed here. Amazing work. Drake being a great member of the lab and doing lots of wonderful things in the lab. And one of his other great qualities is he's actually an excellent mentor to students and other members of the lab. He's really been amazing, such that I don't think he actually touched the scalpel in this wonderful, arguably more beautiful display, but nonetheless was able to guide younger folks through that. The second story that Drake will tell you about really pushes the limits of what we can do in the lab, I think. It's a really impressive, large project that he took on and has led, bringing it to a really impressive point. And I think you'll see that it also melds his interest in quantitative science and biology, which I think is just perfect for him. So without further ado, let me introduce Drake Edwards in his dissertation entitled Novel Understandings of How Cancer Prevents and Responds to DNA Damage. Take it away, Drake.
All right. Thank you, Scott, for those incredibly kind words. Okay, let's get started. First, I want to start with an overview of the motivation for the work I've been doing over the past several years. The fundamental basis for cancer research is summarized here on this slide. Cancer cells are normal cells in our body that have acquired dysfunctional changes which cause them to grow uncontrollably and spread throughout the body, eventually leading to organ failure and death of patients. Because cancer cells are so similar to normal cells, it's a very difficult task to develop treatments that can preferentially kill cancer cells while leaving normal cells intact. Luckily, there are key differences between cancer and normal tissue that have been discovered over many decades of scientific research. Understanding these differences can uncover vulnerabilities in cancer, which can be targeted by cancer therapies. This is a graphic showing some of the hallmarks of cancer which separate the way cancer cells operate in comparison to normal cells. Among them include things like cancer's ability to grow and replicate indefinitely, while escaping the immune system and signals to suppress growth. In addition, one of the hallmarks of cancer that our lab is interested in is that cancer cells have high levels of genome instability. This is because the systems that control both the prevention and response to DNA damage in a cancer cell are different than normal cells. The goal of my research is to understand the mechanisms by which these DNA damage systems are different in cancer and use that knowledge to uncover potential therapeutic targets to more effectively treat cancer in patients. My thesis work revolves around two different projects, which I'll be talking about today. The first is the role that BRD4, an epigenetic protein, has in preventing DNA damage in a cancer cell by preventing collisions between transcription and replication. The second project seeks to understand how the tumor microenvironment, which is defined as the environment which surrounds a growing cancer cell, influences the way it responds to DNA damage caused by radiation. Okay, so DNA, the double-stranded helix we learned about in high school biology, is an instruction manual that contains all the information that a cell needs to build proteins which perform all of the essential functions within a cell. To build a protein, RNA polymerase opens and reads the DNA to create an mRNA molecule in a process called transcription. The mRNA molecule is then shipped out of the nucleus into the cytoplasm, where a ribosome uses that mRNA molecule to build a protein, and that process is called translation. This is the pathway by which all proteins are made in the cell, and the entire system depends on DNA being intact and undamaged. Too much damage to the DNA leads to a cell being unable to perform its functions, and thus preventing DNA damage is essential to cell survival. There are 5 phases of transcription. The first is recruitment, where transcription factors recruit RNA polymerase to the promoter region of a gene. After recruitment, the transcription factor TF2H phosphorylates RNA polymerase on serine 5 of its tail, and this induces transcription initiation. After about 50 base pairs of transcription, RNA pol undergoes a pausing event. Then BRD4 activates CDK9 to phosphorylate serine 2 on the tail of RNA polymerase to release it from this transcriptional pause and allow transcription elongation for the entirety of the gene body. Finally, at the end of the gene, both RNA polymerase and the transcript are released during the termination phase. And so I'm going to be talking a lot about how BRD4 plays a role in this. This is a picture of the cell cycle of an actively replicating cell. During G1, the cell begins to grow and develop cell machinery. During S phase, the cell creates a copy of its entire genome to prepare for cell splitting. Growth continues during G2 phase, and finally the cell splits into 2 daughter cells during mitosis, or M phase. Transcription occurs during each one of these phases of the cell cycle, including during S phase, where both DNA replication and transcription must share the same DNA template. Here's a picture of a DNA replication fork. To replicate the entire genome, replication origins are fired across multiple locations in each chromosome, and DNA polymerase accurately copy each strand of DNA in a 5' to 3' fashion. This process is tightly regulated and controlled to ensure a complete and accurate copy of all of our DNA. Sometimes, however, the replication fork can run into roadblocks, and this is termed replication stress. When replication stress occurs, a complex signaling cascade occurs to signal DNA damage. For example, by phosphorylating histones like H2AX, and that's termed gamma H2AX. And recruit DNA damage response proteins like ATR to locate and resolve the cause of the replication stress. There are several causes of replication stress, including conflicts with RNA polymerase II and the transcription bubble. As I mentioned before, during S phase, both the replication fork and the transcription bubble are using the same DNA template. Collisions between the two are sources of replication stress. DNA damage and even cell death can occur if enough DNA damage accumulates. The cell has developed complex systems to separate transcription, both in time and space during S phase. In this experiment, the transcription bubble is labeled in red, and the replication fork is labeled in green. Throughout the entirety of S phase, the two occupy different areas of the chromatin to ensure that conflicts don't arise. Under normal cell programming, replication forks and transcription bubbles should never collide. However, certain events, such as the formation of R loops, leads to instances where RNA polymerase collides with the replication fork. An R loop is broadly termed for a 3-stranded structure, which arises when RNA binds to a complementary strand of DNA, and that displaces the non-complementary strand. R loops can play physiologic roles, but can also lead to transcription replication conflicts when formed in the wrong place at the wrong time. During transcription, for example, the nascent RNA strand can re-anneal with its template strand as shown here. The R loop can then tether RNA polymerase II to the chromatin and cause it to become a sitting duck for collisions with the replication fork. These aberrant R loops and subsequent transcription replication conflicts, if not prevented or resolved, can lead to increased amounts of replication stress, and then subsequent DNA damage and even cell death. Our hypothesis was that BRD4, which releases RNA polymerase from that transcriptional pause, prevents R loops from forming when the initial RNA transcript and subsequent transcription replication conflicts occur. So let me tell you a little bit more about what we know about BRD4. BRD4 is part of a family of proteins called the bromodomain and extraterminal domain, or BET protein family. As I just mentioned, BRD4 plays a role in transcription and is also known to influence chromatin dynamics. BRD4 has 4 domains. It has 2 bromodomains, which bind to acetylated lysines on histones and allow BRD4 to interact with the chromatin. It also has an extraterminal domain, which is not well understood. And finally, BRD4 has a C-terminal domain, or CTD. The C-terminal domain is responsible for activating CDK9 for phosphorylation of serine 2 on RNA polymerase and release it from that transcriptional pause. In the past decade, inhibitors of the BET family of proteins, such as JQ1, shown here, have been discovered to be effective in treating many cancers. However, the mechanism by which they work is not well understood. JQ1 and other BET inhibitors bind to the bromodomains of the BET family and prevent it from interacting with the chromatin. When I first joined the lab, we noticed that treating cancer cells, such as HeLa cells, as shown here, exhibited a DNA damage response when treated with JQ1. In this experiment, we see that JQ1 treated cells show an increase in gamma H2AX, a marker of DNA damage, compared to those treated with the control. And here the control is DMSO, a solvent in which JQ1 is dissolved. Shortly after the development of JQ1, a new class of small molecule degraders was developed to cause degradation of BET proteins. The degraders, such as DBET6, which I'm showing here, work by linking JQ1 to an E3 ligase recruiter. This results in the BET protein, such as BRD4, being ubiquitinated and subsequently degraded by the proteasome. we saw that dBET6 led to rapid degradation of BET proteins in about an hour and led to a faster, more robust DNA damage response compared to JQ1. We then sought to understand the mechanism by which this DNA damage response was occurring. So first, we wanted to understand whether treatment of cancer cells with dBET6 was caused by BRD4 loss, or compared to one of the other BET proteins. So we developed an inducible BRD4 overexpression system, which could allow us to test whether BRD4 overexpression could rescue the DNA damage effects caused by dBET6. In this system, upon administration of doxycycline, both BRD4 and the fluorescent marker mCherry are co-expressed. Here we see the system at work. On the left is the DAPI signal, which marks the nucleus of each cell. Then we see that cells that are expressing mCherry in red are also expressing BRD4, labeled in green. So we knew that our system was working. So after we figured out that it was working, we tested whether overexpressing the full-length isoform of BRD4, also known as isoform A, could rescue the DNA damage caused by dBET6 treatment. Here we see that the empty vector, deep in the empty vector, dBET6 induces loss of RNA polymerase II activation and a subsequent increase in DNA damage. However, overexpression of isoform A rescues both the DNA damage phenotype as well as the phosphorylation of serine 2 and RNA pol 2. We then wanted to test if a specific domain in BRD4 was necessary for this rescue effect. So we added to the panel a truncated form of isoform A, which is missing the C-terminal domain. And then we also tested a shorter isoform of BRD4 called isoform C, as well as a truncated form of isoform C missing the extra-terminal domain as well. What we saw is that all isoforms missing that C-terminal domain were unable to rescue the DNA damage or the RNA polymerase II activation loss caused by dBET6. With this experiment and others I can't show due to time, we concluded that RNA polymerase activation by BRD4 is really important in preventing this DNA damage in cancer cells. So as I mentioned in the introduction, conflicts between transcription and replication occur in S phase. We noticed that when we treated cancer cells with dBET6, we saw large amounts of damage in kind of a sporadic population of cells that seemed to grow larger over time. Because of this, we thought that the damage caused by dBET6 might be cell cycle specific. To test this, we used a thymidine analog called EDU to label cells that are going through S phase. Cells that are actively replicating will incorporate this molecule and glow green. Simultaneously, we treated with dBET6 and stained for DNA damage for the DNA damage marker gamma H2AX in red. Our hypothesis was that if dBET6 is causing these conflicts between transcription and replication, only green cells would show damage. In fact, that's what we saw. So in this immunofluorescence experiment, we see that EDU stained cells in green are the only cells that display DNA damage signaling after a brief period of dBET6 treatment. This population of cells grew bigger as more cells entered S phase. We also saw other signs that BRD4 was causing replication-specific DNA damage and cell death. Here on the left, we performed a DNA fiber assay. In this assay, we pulsed with IDU to label replication forks in green. Then we treated with dBET6 and did a second pulse with CLDU to visualize how replication forks changed their behavior after treatment, and we marked that in red. We saw that dBET6 halted replication forks compared to the DMSO control. We believe that this is because the loss of BRD4 is causing these conflicts. We also used flow cytometry to quantify how the distribution of cells in the cell cycle changed with dBET6 treatment. We saw that dBET6 treatment led to a loss of cells in S phase and an increase in the sub-G1 population of cells. This finding indicates that cells in S phase are undergoing apoptosis and dying after dBET6 treatment. Finally, we tested whether overexpression of isoform A, the full-length version of BRD4, could rescue the replication-dependent DNA damage caused by dBET6. We again saw that the C-terminal domain of isoform A was necessary to rescue this DNA damage in actively replicating cells. Now that we had established the loss of BRD4 and its or loss of BRD4's ability to activate RNA polymerase II led to an increase in transcription replication conflicts, we then aimed to understand the mechanism by which TRCs were forming. As I mentioned in the introduction, R loops can form when a newly transcribed RNA re-anneals with its template DNA strand, as we see here. And this leads to a tethering of RNA polymerase to the chromatin and subsequent conflicts. Our hypothesis was that in the absence of BRD4, RNA polymerase II is unable to continue with transcription elongation and remains in a persistently paused state after a brief period of transcription. The nascent RNA then becomes more likely to re-anneal with its template in this persistently paused state, leading to R-loop-dependent transcription replication conflicts. One tool that we have to study R loops is RNase H1. RNase H1 is an endonuclease which specifically cleaves the RNA strand of an R loop. We can use the overexpression of RNase H1 to try and determine whether DBET6 is causing damage through an R loop-related mechanism. If the damage is being caused by increases in R loop formation, overexpression of RNase H1 will rescue the DNA damage effect. In this experiment, we use two forms of RNase H1. The first is the wild-type form, and then the second, termed WKKD, is a mutated and inactive construct of RNase H1, which can neither bind to R loops nor resolve them. Each of the RNase H1 constructs was labeled with a V5 tag, so cells carrying the construct are shown in yellow. We saw that expression of wild-type RNase H1, but not WKKD, was able to rescue the DNA damage caused by DBET6, indicating to us that loss of BRD4 is leading to an increase in R-loop-dependent transcription replication conflicts. In addition to the WKKD mutation construct of RNase H1, we also used a different mutation construct, and this one was called D210N. D210N can bind to, but not resolve R loops. We then used this mutation construct to perform R loop chromatin immunoprecipitation followed by sequencing, also known as R-ChIP-seq. Here are the results of R-ChIP-seq as well as BRD4 and gamma-H2AX ChIP-seq. Occupation of the chromatin by BRD4, R loops, or gamma-H2AX compared to input are labeled in red, while absence of occupation is marked in blue. Here we see that across the genome, DBET6 induces both DNA damage as well as R loops. We then used qPCR to verify that R loops were forming at sites of normal BRD4 occupation. We saw that R loops were forming across the gene bodies of genes occupied by BRD4. In conclusion, we showed that under normal circumstances, BRD4 works to activate CDK9 in order to phosphorylate serine 2 on the tail of RNA polymerase II, releasing it from transcriptional pause and allowing for transcriptional elongation. However, if BRD4 is inhibited, degraded, or missing its C-terminal domain, R loops form and tether RNA polymerase II to the chromatin, leading to transcription replication conflicts, replication stress, DNA damage, and cell death. These data are important to understanding the mechanism by which BET degraders in clinical trials work. Furthermore, these findings may lead to a better understanding of which cancers may be more susceptible to BET inhibitors, such as those with high transcriptional and replication roles. So that's the first project. So now, next I'm going to switch gears and talk about the other major project that I've been working on during my time in graduate school. In this project, we developed a novel model system for doing high-throughput genetic screening in glioblastoma. The purpose of this project was to understand how the tumor microenvironment changes the way brain cancer responds to treatment with radiation. Understanding this could allow us to illuminate previously unknown therapeutic targets which could sensitize glioblastomas to radiation treatment. So, glioblastoma is the most common and most deadly primary brain cancer in adults. Average survival is measured in the range of months after diagnosis, with only about 5% of patients surviving past 5 years. The current standard of care for these patients includes a three-pronged approach, including surgical removal of the tumor, chemotherapy with the DNA alkylator temozolomide, and radiation therapy. Despite decades of research, scientists have been unable to find effective therapeutic targets in glioblastoma due to the many challenges involved with studying and treating glioblastoma. Glioblastomas are extremely complex tumors with no prominent driver mutation and carry high levels of heterogeneity within the tumor, meaning that cells that make up the tumor have distinct genetic landscapes from each other, making it difficult to find a single treatment to treat all the different cell types making up the tumor. In addition, glioblastomas are highly invasive, and drug treatments are difficult because they must pass the blood-brain barrier. Finally, the tumor microenvironment plays a large role in how the tumor behaves inside the brain. One of the fundamental problems with studying any type of cancer is the fact that in order to study and research the disease, we must first remove it from the patient and place it in an artificial environment. Historically, this involves putting cancer cells in a plastic dish and bathing them in a mixture of nutrients and animal serum containing hormones and growth factors. This culture technique, termed in vitro study, has been incredibly important to our understanding of cancer because it's simple, it's cheap, and it's scalable for high-throughput genetic screening. However, putting cancer cells in a plastic dish is a far cry from the environment from which it came, leading to gene expression changes and clonal selection. To combat these drawbacks, we can use animal models such as mice to study cancer. This allows for the glioblastoma cells to be implanted into the mouse brain and allowing it to grow in a native brain environment. There are several methods for doing this, but all of them are expensive and don't lend well to high-throughput genetic screening. We aim to find a new method of culturing glioblastoma cells in order to perform a genome-wide genetic screen to find radiation-sensitizing gene targets when cells are growing in a native tumor environment. Through collaboration here at Duke, we hypothesized that using brain slices as a model for high-throughput screening would allow for a more in vivo-like tumor environment while keeping resource costs relatively low. In this model, mouse brains are harvested, thinly sliced, and kept alive on a bed of agar containing nutrients. Cancer cells are then plated on top of the brain slices and cultured there during the experiment. Each mouse yields about 10 slices and drastically increases the scalability of the model compared to in vivo, while still putting cancer cells in contact with normal brain parenchyma. We believe that a genetic screen on brain slices would have much different results than one performed in vitro, and that the results would more closely validate in vivo. To begin to understand what effect brain slices had on the way cancer cells grew, we used a human glioblastoma procured from a patient here at Duke. After removal of the tumor, we took a portion of it and immediately froze it for RNA sequencing, which I'll talk about in a second. The rest we dissociated, labeled with GFP, and grew in one of 4 conditions: standard monolayer culture on plastic, culture in a low adherence environment which allowed for the formation of tumor spheres, inside a Matrigel 3D scaffold, or on brain slices. We then observed the morphological changes each exhibited. And here are the morphologies of each culture method. So as you can see in the inherent culture method, you can see a flat monolayer of cells that become very spindly. In the tumor spheres, you see that they can bunch up on the plastic and form these spheroid tumor-like structures. In the organoid, you see that they're suspended in this 3D scaffold and have long projections. And then finally on brain-sized culture, they form, it's hard to see here, but they form these tumor-like structures on the brain parenchyma and even show some invasion. We then sought to understand the effects that each culture method had on gene expression. We took cells from each culture method as well as a piece of that original tumor and performed RNA sequencing to analyzing which gene pathways were being transcribed in each. Amazingly, we found that among the 4 culture conditions, the brain slices most closely represented the gene expression profile of the original tumor. In this multi-dimensional scaling plot, samples with a similar gene expression profile are closer together. The cells grown on the brain slices, shown here in red, are most similar to the original tumor in black, and we see that because they are closest together in this plot. Next, we wanted to compare specific pathways that changed when cells were grown on plastic, on brain slices, or implanted in a mouse brain. To do this, we chose to use the VMDK mouse model because it is one of the few mouse strains that develop spontaneous high-grade gliomas, of which the SMA 560 cell line is derived. Using this model allowed us to not only grow the SMA560 cell line on plastic and on slices, but also allowed us to implant the tumors into immunocompetent mice for study, as shown here. Here we show the different morphologies of SMA560 cells grown in the different model systems. Cells grown on the brain slices invaded the normal tissue and formed tumor-like structures, similar to how cells grew inside the mouse brain. And then again, you can see that the cells grown on plastic really spread out in a thin monolayer on the plastic, which is different than what we expect for a tumor grown in a patient. We then performed RNA-seq on the 3 model systems and compared gene pathways that were upregulated in vivo or on slices compared to in vitro or on plastic. Here we see that pathways involved in the extracellular matrix, biological adhesion, and the cell surface were upregulated when the tumor cells were cultured in vivo, so inside the mouse. Similarly, when SMA560 cells were grown on brain slices, we saw that the same pathways were upregulated to when the cells were grown in vivo. Those marked by the red arrow here were also upregulated when the cells were cultured in the mouse brain. So the cell surface, the extracellular matrix, and biological adhesion. When we compared the cells grown in all 3 model systems and looked at which pathways were co-upregulated on brain slices and in vivo compared to in vitro, we saw that, again, the extracellular matrix, biological adhesion, cell surface, all kinds of things that indicate that the cell is interacting with its tumor environment, all those pathways were upregulated when the cells were grown either on brain slices or grown inside the mouse. So as I mentioned previously, radiation therapy is a staple in the treatment of glioblastoma. Radiation therapy works by inducing lots of DNA damage to cells. Because cancer cells have differences in the way they respond to DNA damage, they are more sensitive to radiation than most normal cells. However, one of the limitations of radiation therapy is that it's hard to effectively treat all the cancer without some of the normal tissue also exhibiting lasting damage. Thus, finding a way to sensitize cancer to radiation therapy increases our ability to treat these cancer cells while leaving normal tissue unharmed. So much work has been done to increase our understanding of the DNA damage response to radiation. I'm going to highlight one part of the DNA damage response here. DNA PK is encoded by the PRKDC gene, shown here, and is part of the response to radiation. When radiation induces a double-strand break in the DNA, DNA PK is recruited to the site of damage and activates the non-homologous end-joining pathway, which joins the free ends of DNA caused by the radiation back together, fixing the break. In the context of glioblastoma, cells that are missing the functionality of DNA PK are unable to repair those breaks as effectively and are much more sensitive to radiation. To test whether specific genes are important to the radiation response in glioblastoma cells, we used a technology called CRISPR. CRISPR has been developed as a 2-part system in which we can target genes to become non-functional. The first part of the system is the sgRNA, so shown here. The sgRNA specifically binds to the DNA of the gene to be targeted and recruits the protein Cas9. Cas9, upon being recruited to the target gene, makes a cut in the DNA leading to a mutation, such as a frameshift mutation. This mutation leads to a jumble of the genetic code, resulting in a totally non-functional protein. We use this strategy to knock out individual genes to understand their importance in a cell's survival following radiation. To test whether brain slices could accurately replicate the radiosensitization effects seen in other models, we used CRISPR-Cas9 to knock out the DNA PK gene in SMA560 cells. And then we irradiated them and measured cell survival. On the left, we can see that CRISPR was effective in knocking out the DNA PK gene. And then on the right, we see that on plastic, as we expected, loss of DNA PK led to a decreased cell survival when irradiated compared to a guide targeting a control locus, AAVS1. We then performed the same experiment on brain slices and saw that loss of DNA PK was also highly radiosensitizing in this context. This gave us confidence that doing a genome-wide screen in which we individually knocked out each gene in the genome would be possible on the brain slices. So, Given the ability of brain slices to provide a more in vivo-like environment for the SMA 560 cells, as well as identify those known radiation sensitizers, we designed and performed a genome-wide CRISPR radiation screen to observe how growing cells on the brain slices would have on the response of the glioblastoma cells to radiation. Here's the step-by-step process by which we did that. First, we infected the SMA 560 cells already expressing Cas9 with a genome-wide CRISPR library called GECKO. We then used puromycin to kill all the cells except for those which were properly infected with an sgRNA. Next, we split the cells into two groups. One group was grown on plastic in vitro, while the other was grown on the brain slices. Then for each culture method, we either irradiated the cells or didn't. And then after 5 days, we harvested the genomic DNA from the cells and used next-generation sequencing techniques to identify which sgRNAs were in cells that survived. We then compared which cells survived in the irradiated group to the unirradiated group in each growth condition. sgRNAs that targeted genes important to radiation survival would be represented in the unirradiated group, but would be almost absent in the irradiated group because all of those cells had died. Okay, so here are the results of the screen. On the left, we see that there were 949 gene targets that sensitized cells to radiation when grown on plastic. Additionally, there were 828 gene targets that sensitized cells to radiation when grown on the brain slices. And then finally, there were 33 gene targets that were sensitizing in both conditions. Assuredly, DNA PK knockout, which we did mention earlier, was one of the most important radiation sensitizers on plastic and the most radiation sensitizing gene knockout on slices. We then looked at specific pathways that were particularly important to the radiation response in each screen, as well as the overlap between them. Here I looked at the top 100 radiosensitizing genes in the screen for cells grown on plastic and asked which pathways those genes were part of. As we expected, most of the pathways involved were part of the classic DNA damage response, including double-strand break repair, DNA repair, and response to ionizing radiation. We then looked at genes that were radiosensitizing in both screens. Again, we saw that pathways were the pathways that were involved in non-homologous end joining, such as DNA PK, double-strand break repair, and response to ionizing radiation were all important for cells grown both on plastic and on brain slices. And then finally, we looked at the top 100 genes that were radiosensitizing on brain slices. Amazingly, we saw pathways that were not only different than the ones that were important to the radiation response on plastic, but pathways that were also upregulated, as we saw earlier, when cells were grown on brain slices or when implanted in the mouse brain, such as cell morphogenesis or the regulation of multicellular organismal processes. So here's a global picture of all the gene ontology terms that were enriched in each screen. As shown in the key on the left, each node is marked with a color. If the node is red, it is a pathway that was important to radiation survival on brain slices. A blue node was a pathway important to radiation survival on plastic. Finally, nodes that carry both colors were important to both screens. The size of the node represents how many genes that pathway contains, and links between nodes represent related pathways. The closer two connected nodes are, the more similar they are. Tight clusters of nodes are most likely to be truly part of pathways involved in radiation survival. And then, so here we can see there are a variety of clusters, and some containing both colors, as seen here. Both the DNA damage response and the response to radiation were important pathways for radiation survival in both screens. This is reassuring, as both screens were able to identify known radiosensitizing genes. We also saw clusters that were almost all blue, representing that these pathways were only important for radiation survival when cells were grown on plastic. The response to hormones, for example, may only be important to the radiation response when cells are bathed in artificial media containing animal serum. And then finally, we saw clusters that were almost all red, illuminating pathways that were important for radiation survival when cells were grown on brain slices. Interestingly, we saw pathways that were also upregulated in the RNA sequencing data I mentioned earlier in the talk, such as cell morphogenesis. We also saw clusters involved in a cell's interaction with its environment, such as the regulation of vasculature or the regulation of synapses. This is exciting data because this screen has uncovered new potential therapeutic targets which could sensitize glioblastoma to radiation. Furthermore, it provides a novel screening platform which may more accurately illuminate true gene targets that would help human patients. The work does not stop here, however. The next steps in this project are to validate several of the targets in an actual mouse. We developed a custom limited CRISPR library to test the validity of these hits across all 3 model systems, in vitro, on slices, and in vivo. We expect that gene targets found on the brain slices will more likely validate than those we find on plastic. In summary, these 2 projects shed light on how cancer both prevents and responds to DNA damage, increasing our understanding of how cancer works and how it can be targeted. In the first project, we found that the transcription regulation activity of BRD4 plays a critical role in preventing collisions between transcription and replication during S phase. In the second, we found that the tumor environment really changes how glioblastoma responds to radiation. I'm so proud to have had the privilege to work on these projects and contribute in some small way to the fight against cancer. But it takes a village to do this work, and I would not be sitting in front of all of you today if it weren't for a massive group of friends, family, and colleagues. First, I'd like to thank current and former members of the Floyd Lab, specifically Rohin Maganti, who was just awarded the prestigious Goldwater Scholarship and helped me with both projects. Jared and Jie, who helped me so much in the first project, as well as Denise Dunn, who sliced enough brains to make her head spin when we were working on the second project. And finally to James and Elena, who made the lab run during my time here. I'd like to thank Steve Keir and Peter Fetchy in the neurosurgery department, who provided the human tissue samples and gave me space in their lab to perform the tumor implantations. I'd like to thank my thesis committee for taking the time out of their busy schedules to mentor and support me all these years, as well as the Duke MSDP for taking a chance on me and providing me with 7 years of support. I'm so grateful for you all. Next, I'd like to thank my mentor, Scott Floyd, who took me into his newly started lab and gave me opportunities to fail, learn, and grow as a biologist. He encouraged me to expand myself as a scientist, whether it was attending a bioinformatics course at Cold Spring Harbor or advocating for science funding in Washington, D.C. I'd like to thank my friends, family, and the med school for providing me opportunities to maintain life balance, whether it was performing in med school musicals or climbing extremely tall things and overcoming life's challenges. I'd like to thank my family, specifically my parents, who have always given me the opportunity to be curious about the world, even when my chosen path wasn't the most straightforward. And my 3 younger brothers, who have always inspired me to be the best person I can, both in my career and as a person. I'd like to thank our 2 puppies for endless snuggles and support on hard days. Charlie on the left, who we've had for about 5 years, and Tess on the right, who just joined our family last week. And finally, thank you to my kind and brilliant wife for all of her support and willingness to adventure through life with me. I would not be where I am today without her. You keep me inspired to move forward on the hardest of days and keep me humble on the best of them. I love you so much. Okay, so with that, thank you for listening, and I'll take a few questions. Okay. All right, so let's see if we have some questions. All right. Okay, so if anyone has questions, go ahead and put them in the comments. We'll see if we can get it. 1 or 2 here. Okay. Let's see. Okay. So we have a question from Amanda. The question is, did you confirm that sub-G1 cells after DPET6 were apoptotic versus senescent or another mechanism? That's a great question. And yes, we did test that. So we tested that using apoptotic markers such as cleaved PARP. And we did see an increase in cleaved PARP. So yes, we did see that as well. And we saw that cells treated with DBET6 over time tend to die and float off the plastic. Let's see. Okay, we have a question from Jacob. Okay, the question is, a key feature of the tumor microenvironment is likely the biomechanical cueing. How do you think that kind of mechanotransduction might play a role in vitro versus the brain slice? So I think it plays a really important role. I think that cancer cells take a lot of cues from their environment. And when a cell is growing in vivo, it's surrounded by all sorts of things: the microglia, neurons, other supporting cells, vasculature, etc. And I think that the signaling between the cancer cells and the natural brain parenchyma plays a really important role. So I think that when cells are grown on the brain slice, we still get a lot of that as well, compared to plastic, where they're not really getting any sort of feedback from supporting cells. Okay, so we have another question from Amanda. She's asking whether I can speculate on why adherent GBM cells were more similar to in vivo and slices than the tumor spheres or organoids were. Amanda, that's a great question, and one I don't have a great answer to, and something I would like to explore further. You might think that tumor spheres that are putting all the cells in a similar context, in a big ball of cells, might be closer to the in vivo than grown just on a monolayer on plastic. And other groups have shown this. So I'm not sure why our specific human glioblastoma didn't show that similar phenotype. Same with the matrigel. I think in that chart we saw that the matrigel organoids put the cells way out on the edge. In our hands, we've seen that cells grown in this way have a harder time replicating, and so I'm not sure if that has something to do with it. And we're constantly working to try and improve our ability to make efficient organoids and grow tumor cells in that environment. Okay, I don't see any more. I just want to say thank you once again to everyone that was able to come out. I love you all and hope to hear and see you all soon. All right, bye everyone.